Hi there. So the topic I'm going to be talking about is congenital Zika syndrome. Before I talk about that, I need to talk about the Zika virus itself. So the Zika virus is a mosquito-borne filovirus. The species of mosquito most commonly known for carrying this disease are Aedes africanus, Aedes aegypti, and Aedes albopictus. Infected people can transmit it to other people through the exchange of body fluid, i.e., it can be sexually transmitted. Symptoms of the virus include fever, rash, arthralgia, conjunctivitis, myalgia, headache, retroorbital pain, edema, and vomiting. Um, these terms might be new to some of you, so I'll just go over them quickly. Arthralgia is joint pain. Conjunctivitis, just a fancy word for pink eye. Myalgia is muscle pain. Retroorbital pain is pain in the back of the eye. And edema is the fluid buildup in tissue or body cavities. The virus is diagnosed through blood tests. So two things are screened. So firstly, viral nucleic acids are screened using reverse transcription polymerase chain reaction. And secondly, they screen for IgM antibodies using IgM capture enzyme linked immunosorbent assay, or MAC-ELISA for short. Currently, there's no vaccine available, although once a person recovers, they're generally immune for the rest of their lives. Now moving on to congenital Zika syndrome. So if a pregnant woman gets the Zika virus, she can potentially pass it down to her fetus, which results in congenital Zika syndrome. If the mother is infected within the first trimester, there's about a 90% chance the fetus will be affected. And the risk continues into the second, third trimester, although it is significantly lower after the 10th week. Congenital Zika syndrome is characterized by microcephaly, which is the abnormally small head of a baby, which can be seen in the first picture. This is usually due to the brain tissue not developing properly and just the brain not developing properly in general, which is the brain anomalies. Central nervous system damage, which is similar, like the cells don't grow properly. And there's also calcification of brain tissue and nerve tissue. Then ocular anomalies, so it can affect all parts of the eye. However, a particularly bad thing is that you can get calcification in the optical nerve, which is obviously not good. And lastly, congenital contractures are the improper development of bone and joints, which cause these twisted and different sized bones that can be seen in the bottom figure here. All right, so the congenital Zika syndrome varies in severity. However, most oftentimes, it results in death. And then those that do survive have severe motor and cognitive disabilities, which again often leads to death in the long term. Also, I'd just like to say there's very little information on the long-term effects of both Zika virus and congenital Zika syndrome, because it's relatively, like, research has only started relatively recently, since the 2007 outbreak. So yeah, there's very, very little that's actually known about it. So the paper I'm going to be talking about is discordant congenital Zika syndrome twins show differential in vitro viral susceptibility of neural progenitor cells. All right, so what kind of inspired this research? So firstly, congenital Zika syndrome only occurs in about 6 to 12 cases, percent of cases where the pregnant mother is infected by the Zika virus. And secondly, neurodevelopmental dysfunction of congenital Zika syndrome was shown to be caused by impairments in neural progenitor cell growth and survival. So these two things inspired previous research, which showed that neural progenitor cells could actually respond differently when infected with Zika virus. So they can respond in different ways. So with this study, the aim of this study was to use discordant twins for the genetic contribution determining the fetus's outcome of gestational infection. Discordant twins means that one of the twins was affected by the condition, and one of them was totally fine. So to achieve this, what the study looked at was the in vitro neural progenitor cell expression profile and outcome of the Zika virus infection in human-induced pluripotent stem cell derived neural progenitor cells from the discordant twins. That was a mouthful. All right, so the subject they used, they used 2 pairs of monozygotic twins, both of which were concordant. So concordant means that both of them were affected. Then they used 7 pairs of dizygotic twins, 6 of which were discordant, which means that one of them was affected, one of them wasn't. And then one of those 7 was concordant. And then they also used 10 unrelated babies that all had the condition. So the first test they did was basically to look at the inheritance pattern of the Zika virus. They looked at the baby's genome, and actually they found 8 genes associated with microcephaly, which again is the small head. However, the data wasn't statistically significant, so that's why I didn't put it here, it wasn't really used. Anyways, what they did determine is that congenital Zika syndrome is not inherited in a dominant manner. They didn't determine how it is inherited, but they just know that it's not a dominant manner. The second experiment they did was the generation and characterization of HIPSCs, which are human-induced pluripotent stem cells. So they did a bunch of experiments on them. But pretty much the results showed that there was no genetic differences between the affected and healthy individuals. And I'm going to come back to this result later on. So just keep that in mind. The next test they did was computed tomography scan or CT scan of the discordant twins. The results of which are shown here, of the twins' brains. The affected babies obviously had the typical abnormalities seen in this condition, which are the second and fourth column. And then surprisingly, actually, the unaffected babies show no visible alteration. So the condition completely didn't have any effects whatsoever. And then there's also a finding where they found brain calcification in affected babies, which we already know, but they're pointed out with these white arrows. All right, next thing was the in vitro growth of neural progenitor cells when infected with the Zika virus. So the way they tested this was by infecting both affected and unaffected neural progenitor cells with the virus in different concentrations. So the results showed significant reduction in growth and proliferation of affected cells when they were infected with the Zika virus. However, which can be seen in this in the first figure, by the way. So the third column, you can see that the affected cells had much less growth than the unaffected cells, both in the presence of the Zika virus. But what was surprising is both the affected and unaffected cells had very similar growth when no Zika virus was present. So this kind of shows that the babies with this condition could potentially develop normally if they're not exposed to the Zika virus. The bottom 2 figures just represent this in a graphical way. The next experiment was very similar to that previous one. However, in this one, they tested for the virus in the cells instead of the NPCs. So the results were actually very interesting because they showed that cells from an affected individual are more susceptible and better suited for hosting the Zika virus infection. The first figure here shows the number of NPCs that grew relative to the amount of virus in a variety of conditions here. The red line here represents the affected NPCs, while the top line represents the unaffected one, the top black line, not the bottom one. The bottom one is just a control. But yeah, you can clearly see that there's way more virus in the affected cells. Then down, if we look below, MFI, by the way, means mean fluorescent intensity, and that's how they calculated the amount of the virus. You can just see that the affected cells had much more virus in them. All right, lastly, the last experiment, they did an RNA sequence analysis of NPCs. So they found that there were 64 differentially expressed genes between the unaffected and affected cells, affected NPCs. Um, Notably, the gene DDIT4L, which is a known inhibitor of the mTOR pathway, was found to be in a 12.6-fold decrease of mRNA expression in NPCs obtained from the affected individuals. This indicates that the mTOR response is actually an important factor for the successful Zika virus infection. The fact that these results, so I remember I mentioned the other results previously that were important. So they differ from this because the other one they didn't see a difference between the two groups, and this one they did when they analyzed the NPCs versus the stem cells. And so this kind of shows that that the differences only in expression only appear after differentiation. All right, so what do the results of this paper mean? So the fact that both monozygotic babies and one dizygotic baby were concordant, and the other 6 dizygotic were discordant. So this pattern suggests the genetic basis for congenital Zika syndrome susceptibility, which is what led to this research. And the 3 main findings of this research. So firstly, NPCs from affected and unaffected groups had similar growth patterns before Zika virus exposure. This suggests that affected babies would not develop symptoms if they're not exposed to the Zika virus during pregnancy. So secondly, differences in gene expression indicate that the symptoms are a result of altered pathways such as the mTOR pathway. Lastly, no single gene was associated with this disorder. So this suggests that the disorder is a result of multiple factors and not just one genetic genetic contribution, I guess. All right, so lastly, future directions. So this study confirmed the influence of the host genome in congenital Zika syndrome. So this shed a little bit of light on the molecular pathology of the condition and opens the possibility for drug development that would target specific pathways, like the mTOR pathway, and hopefully be able to eliminate the symptoms of the syndrome entirely. Anyways, guys, thank you for listening. These are my references right here. Cheers.